Hello and welcome back to Science of Medicine. Today I want to talk about the optics of vision, physics of this. And in order to do that, I need to tell you a little bit about eyes. Now, vision is a special sense that involves the detection of light rays in the back of your eye and then digitizing this, if you will, processing it in your occipital lobe in the back of your brain so that you can make sense out of your surroundings. And if you look at a person's eye, you'll notice several things. Certainly the eyeball or the globe is in a socket and that there are lids, both an upper as well as a lower lid. And the white portion of that eye, or the sclera, is on the outside. I then have a colored portion, which is called the iris. And in the center, I have a pupil. Now, pupil is just a hole that's going back into the optical system of the eye. And it can change diameter as under the influence of the parasympathetic and sympathetic nervous systems, as well as in adaptation to the amount of light that is present. If I look at the eye from the side, I find that it's a very complicated structure. So if I look at the eye from the front, as I'm having light go in, the first set of transparent cells that I have is comprising the cornea. And the cornea is the first part of the eye to allow for refraction. So I'm going to be able to actually help focus an image at some distance from my eyeball onto the back of my eye. And the cornea is going to be the first thing that does that. Now, the cornea is on top of what's called the anterior chamber. Now, normally, the anterior chamber will not have other cells. It has a gel-like substance, an aqueous humor that we have in there, but it normally wouldn't have cells. Under some pathologic conditions, you can get cells like white blood cells or red blood cells in there, and they fill up, and they're confined to this anterior chamber. As I go back a little bit further, I'll encounter the iris. Now, the iris is the colored portion of our eyes, and that is hereditary. And in the center, I will have a pupil, which is an aperture, an opening for this lens system. And that opening, as I mentioned, can vary based on inputs from the sympathetic or parasympathetic nervous system as well as from light. Immediately behind the pupil is my lens. And my lens has proteins that will allow me to focus the light on the back of the eyeball. And this has the greatest refracting power as I'm going through my eye. And then I'll go into the globe proper, if you will, into the back of the eye, which also has a viscous liquid in it, the vitreous humor. And ideally, what I will do is focus an image on the retina. Now, there is a special spot or very small region called the macula or even the fovea that has the highest resolution, the highest acuity when I focus images on there because it has a very high density of cells that are connected to individual neurons. Now, ideally, this focusing system from the cornea all the way down to the fovea or the macula is going to allow for a perfectly sharp image to exist there. And that will have to be based on a changing of the lens power of the lens system, because I'll want to look at an object that might be 10 inches away or let's say half a mile away and want them to both be in focus. If I look at the back of the eye, I'm using a fundoscope, a special way in which I can actually view the back of the eye. Here is a picture of a normal fundus, and I want you to notice several things. First off, this sort of darkened spot right here, which is the fovea in the center, the macula right surrounding it, is where I ideally like to actually focus my image. Now, I also have off to the side a white disc. I actually have sort of a disc inside of a disc. It looks like there's a disc and a cup in the center. But all of this white area is actually the optic nerve. Now, one of the cranial nerves that you have is an optic nerve, and that optic nerve will then send signals from the neurons that are connected to the specialized cells in the back of my eye, and that will go to the occipital lobe in the back of your brain. You'll also see lots of blood vessels. The larger ones are veins, and the slightly smaller ones are arteries that you'll see that are throughout this time. And this is a sort of normal distribution of those blood vessels. There are things that can look abnormal. For example, if I focused in on the optic disc and found that it was sort of almost obliterated, it was very blurry, and even the blood vessels as they were going over that optic disc would be blurry. This is indicative of papilledema, which is edema or swelling that actually causes this optic disc to actually show up in this fashion. Many times people have retinal damage from diabetes mellitus, and that can cause what we call a diabetic retinopathy. In fact, this is what we will see. One of the first things that we can see from diabetes is this retinopathy, and that's why diabetic patients will try to go every year to their eye doctor in order to actually look for this. But you can see this proliferation of blood vessels, and this is not a healthy situation. And you can also see some areas in white here and here of ischemia where the cells have died. All right, so let's talk a little bit about how the eye is going to process that light. So if I look at the fovea or the macula or even somewhere on the periphery, light rays are going to be focused onto a layer, and that layer is actually going to be of neurons, and they're transparent, and they'll allow for the light to go through to certain sets of cells that will absorb the light. Now, I have two sets of cells. I have what are called rods, which function in dim light, and they detect the presence of any light, irrespective of wavelength, visible light. And then cones, which are functioning more in bright light, and they're specific to detecting one of my primary colors that I can see, so red, green, and blue. And when that light is absorbed, that photon of light is absorbed, there's a conformational change that occurs that produces an electrical signal that is detected by the neurons and then transmitted to the optic nerve, and then from there to the brain for construction of this image. Now, if I looked at a pictorial representation of various cone cells in the back of the eye around the macula, I would see here on the left one that would show cones that would be distributed for green and for red and for blue. Or if I had a person that was colorblind and didn't have the ability to detect red, then I would see an absence of those cells. In the macula and the fovea, I don't have a lot of these cells connected to a single neuron. I have just, unlike the periphery where I do have a number of cells that are connected to the same neuron. And so consequently, I can try to detect that light, that signal, if you will, getting above a certain threshold in my peripheral vision, but I may not have the same good resolution that I do if I actually focus the image back onto the macula. Now, since my eyes have to focus that light and that image on the fovea, I have a set of muscles that will move my eyeballs in a coordinated fashion so that I can then get that light to be focused back at the right place. Now, once I have that signal, you might think that it would be fairly straightforward. It gets transmitted back to the back of the brain, and then I can make sense out of it. But it turns out that that's not the case. It's actually a more complex pathway. It turns out that the neurons that are for the two quadrants that are on the nasal side of each eye, in the center towards your nose, that those actually will then cross over, whereas the outer two quadrants, will actually just go outside and go around. It is the way that we're made, and so consequently, I have part of the signal that crosses over and goes to the opposite side of the brain, and part of the signal that just stays on the same side. This actually is very important. It's very important for a couple reasons. Number one, it turns out that crossover is right about where your pituitary gland is. It's at the optic chiasm. And so if you have pituitary tumors, for example, they can have adverse effects on the eye because it's actually causing some sort of distortion, some sort of pressing on these optic nerve crossings, and that will create a problem. Secondly, it also shows you that the way in which we're processing an image, although you might think about it as being a whole image, is actually broken up into different pieces. And sometimes you can have strokes, you can have areas of neurons that have died along this pathway, and you can actually have like part of your vision, just an outer quadrant or an inner quadrant that might actually be affected by this. All right, so let's talk more about the optics of vision, and before I do that, I want to make sure that you understand some basic ideas. So if I have a converging lens and I have light rays that come in, they will bend towards some focal point that I would have here, a distance of focal point. And remember that that focal point can change based upon the lens and the refracting power of that lens. This is a positive or a converging lens. There is also a diverging lens where I have a focal point that lies in front of that actually causes the light rays to actually spread apart from each other. And so just so that you remember what those two different lenses are. Now in my eye I have a converging lens, but when I have corrections to my vision, I might need to use either of these lenses. And remember that there is a relationship between the distance that an object is from the lens and the real image that is on the converging lens on the other side of the converging lens. It's inverted, but it is still nonetheless a real image. And that relationship is given by the lens equation. So that says that the reciprocal of the focal length, which we call the power, and we give that in what's called diopters. So if you look at your lens prescription on your eyeglasses, you'll see -2 or +2 diopters or something like that. That's just the reciprocal of the focal length in a particular unit. And so this says that the distance between my optical lens and the object, and we'll call that S1, the reciprocal of that, plus the reciprocal of the image, the real image, and the distance to the lens is the sum of those two reciprocals is the reciprocal of the focal length. So what that tells us is if I have an object that I'm trying to focus on, that object is 100 feet away, then in order for me to focus that image at a certain point on the back of my retina, I have to change my focal length. The process by which I do this is called accommodation. Now, the way I'm going to do that is I'm going to change the shape of my optical system. I'm going to change my lens and the shape of my eye. And so what I will do is I will be able to make my eyeball sort of fatter, or my lens fatter or more spherical, or I can make it more flat. So I have a set of muscles called the ciliary muscles that actually are attached to that lens so that they can actually stretch it out, or they can actually make it so that it's more spherical. It turns out that if you look at the retina, all of the refractive indices of the anterior chamber plus the globe itself, as well as the lens and the cornea, the total refracting power of a young person's eyes is generally about 59 diopters. And I can change that quite a bit, to accommodate for seeing things at distances are very, very close. At around age 40 to early 40s, you actually have crystalline changes within the lens proteins that make it so that you can't do this deformation of the lens as easily as what you were able to do when you were younger. And that leads to a decrease in your ability to refract. And you go from about 59 diopters. If you look at the lens, normally you would go about 14 diopters, and then it goes to about 2. And it's a fairly precipitous drop across that time. And then it goes to about 0 by the time you're in early old age, about 70. And that process of the lens proteins changing and leading to a lack of your ability to accommodate is called presbyopia, which is why many times people, when they are in their early 40s, and even if they've never had glasses, they may need to get reading glasses, for example. All right, now there are some lens type imperfections that I want to mention that can be pertinent for what we're talking about. The first imperfection that can exist is that a lens might actually have a slightly different focal length in one plane versus another. So if you see the blue rays are focused at one point and the red rays are focused at another point here, blue and red not being colors, they're just in this image, it's not the actual color of the light. And what that leads to is what's called astigmatism, which we'll talk more about. So if I have a different ability to actually focus the light because of the shape of this lens, and it's in different planes, that leads to an astigmatism. Now I also will have, whenever I refract light, the focal point will differ depending upon what wavelength. We know that the longer wavelengths, such as the reds, are refracted less than the shorter wavelengths, such as the blues. And that's called chromatic aberration or color aberration. And for us, we don't have a way of really accommodating for that. Our mind actually corrects this, but the lens system will not do that. So let's talk about some typical problems that occur with vision. So by far, one of the most common optical problems that the eye has is nearsightedness or myopia. This is a heritable trait, and so if your parents had myopia, odds are really good that you will also have this also. In myopia, the image is focused when the lens is relaxed. In other words, I'm not making it spherical, in front of the retina, in front of the fovea. And so what that means is by the time you get it back here to wherever it is, up to wherever the retina is, it's going to appear blurry because it's not going to be in focus. There's no real way for the lens system to try to push that backwards. And so you have to actually correct this by putting a lens in front of the eye to be able to do this. And we actually will add that refracting power, or add that ability to actually move that point backwards to where it's supposed to be by putting lenses on both the right side as well as the left side when you wear glasses or contacts. And we signify this in a special nomenclature. Now, if I'm referring to the right eye, oculus dexter, I'll say OD, and if I'm referring to the left side, I'll call it oculus sinister, going back to archaic kinds of things where people thought that left was somehow bad. And I also have the reciprocal of the focal length, which we said in terms of the power here, which is in diopters. Now, the minus is referring to the fact that this is a person that has a correction that is myopia. We'll have another one where the image will be formed in a different location, and we'll actually have a different sign on that. So, if you have a prescription for myopia, and your OD is -2 diopters, and your OS is -1.50 diopters, then that means that you have myopia, and that your right eye needs a stronger refracting power in order to get it corrected. So it's worse. Your myopia is worse in that right eye. Now, sort of the opposite of that is hyperopia, which is farsightedness. And in this case, the image is focused on the back of the retina when I've relaxed my ciliary muscles and I've not deformed my lens. So at this time, I can actually make adjustments to my vision, to my globe, if you will, to be able to bring this image forward. And so I have some ability to accommodate this by the way in which I actually adjust my lens system here. But I normally will do this by virtue of putting another lens in front to be able to do this. And this hyperoptic correction is also given as one of the focal length or the power in diopters. But now you see there's a plus sign in front of these, recognizing this is hyperopia. So in this case, this person's hyperopia would be worse in their right eye than in their left eye. But I also mentioned to you about the possibility of astigmatism, which is also fairly common as a defect. So remember, if I have astigmatism, I'll have, in the shape of the lens, it will be such that I'll have two focal points. One focal point in one plane and another focal point in another. And I have to correct that with a cylindrical lens in front. And I have to say not only what the power, the refracting power of that cylinder is, but how much off axis is it? Is it 90 degrees? Is it 40 degrees? Where is that? And that is then given by the prescription of cylinder and axis, it'll say. So in this case, +2 diopters on the right eye at an axis of 90 degrees or 1.5 on the left, an axis of 10 degrees, to be able to put both of those focal points back on the fovea. So I hope this has given you an introduction and a little bit of an understanding of this special and remarkable sense of vision. And I'll be here to talk with you about other things soon.